right, biologists, let's take a look at carbohydrates and lipids, theme B, form and function, topic 1, molecules. So chemical properties of a carbon atom allowing for the formation of diverse compounds upon which life is based. So carbon atoms, here we have a model of an atom. It has 6 protons, 6 neutrons, and 6 electrons. Now, we are not going to go too deep into the chemistry side of things, but electrons are arranged according to how much energy they have. If they have more energy, they go on outer layers. If they have less energy, they go on inner layers. So the first layer here supports 2 electrons, and the outermost here, which is called the valence shell, in this case has 4 electrons. Now, if you also take chemistry, you know things are a bit more complicated than this, but for now, we are going to say that the carbon of an atom feels more comfortable with 8 electrons rather than 4. So to achieve this stability, we have to make covalent bonds. You could have an atom of hydrogen here, for example. Let's make it pink with proton. So hydrogen has 1 proton and 1 electron. And now they can share these 2 electrons. So now this atom of carbon has 1, 2, 3, because they are sharing this electron, 4, and 5 electrons in its outer layer, in its valence shell. So you can do that. And now this atom of carbon has 5, 6, 7, 8 electrons around it. So it's more stable. It has the 8 electrons it wants. This is the same as CH4, methane. And the thing is, you don't need 4 different things around this carbon. You could have double bonds, for example. That's the case of CO2, for example. You have carbon, double bond, oxygen, double bond, another oxygen. They are sharing electrons here in each of those bonds. So each of the oxygen atoms is sharing 2 electrons with carbon, and carbon is sharing 2 electrons with each of these oxygen atoms. And you can have carbon atoms sharing an electron with other carbon atoms. And then we have open chains or even cyclic structures. And now we have this atomic, this chemical Lego brick that can create huge molecules and very diverse molecules depending on how we arrange things. So take a look at these molecules. They are identical. They are 2 copies of the same molecule. We have here 2 molecules of glucose. You don't see the carbon atoms except for this one, but they're here. Every time you have an angle like this, it represents a carbon atom. So you have 1, 2, 3, 4, 5, and 6 up here. Carbon atoms, same thing on this side, 1, 2, 3, 4, 5, and 6. So each of these glucose molecules is what we call a monomer. A monomer is a molecular Lego brick that you can use to build even larger molecules. These larger molecules are called polymers. When you have polyester, that's a polymer. That's a huge molecule made of many esters. And in biology, we also have polymers. We already saw one type of polymer. We saw DNA and RNA. Nucleic acids, like DNA and RNA, are polymers of nucleotides. You have nucleotides, which are molecules themselves, but you connect many of them and you can have an RNA molecule, which is a different molecule made of smaller molecules. Its RNA is a polymer and the nucleotides are monomers. So to produce these macromolecules, in this case a carbohydrate, to make this polymer, we use a type of reaction called condensation. And we are going to condense 2 glucose molecules by removing one H2O. We are going to remove 1 molecule of water. How do we do this? Well, here we have O and H, O, H. And if you look close enough, we have H2O. That's a water molecule. You can remove a water molecule here. So what actually happens is we break this bond between the oxygen and the carbon, and now the oxygen needs to make another bond because it lost the bond with the carbon. So it makes a bond here with hydrogen. Hydrogen cannot make two bonds. So it has to break the bond with the oxygen here. So now we have our H2O molecule. That's too dark. Now we have two problems. This oxygen here, this oxygen wants to make two bonds, but it was making one bond with a carbon, one bond with hydrogen, but it lost the bond with hydrogen. This carbon wants to make four bonds, and one of them was with the oxygen, but it lost the bond with oxygen. So we have two atoms here that need to make a bond, and they're close to each other. Well, we can make a bond between them. We can make a bond between this carbon and this oxygen, and that's exactly what happens. Remember, this is a carbon here, and we have the oxygen making the bond with the carbon. Everyone is making all the bonds they need to make, and we still have this H2O that was removed before. Now, what if we want to digest this polymer? What if I want to take this molecule of maltose? What if I want to take this molecule of maltose and break it into two molecules of glucose? I can do that in my intestine. I have an enzyme called maltase, and the way maltase works is by reversing condensation. It's a process called hydrolysis. So instead of removing a water molecule, we are going to add the water molecule. We can add OH to one carbon and the H to the oxygen here. And now we have the two glucose molecules. Form and function of monosaccharides. Monosaccharides are carbohydrate monomers. So mono comes from one, and saccharide comes from the Greek word for sugar, which according to Google Translate is
Zachary.
So monosaccharides, one sugar. They usually follow this formula CnH2nOn, and from here we can see why they are called carbohydrates, because you have one carbon for every H2O, for every water. Water, carbohydrate. And this N can be a number between 3 and 7. Most commonly, we're going to see 6 for glucose and fructose, for example, or 5. Like in this case, we have a carbon here, and this one up there. So we have 5 carbons. This is ribose from RNA. Ribose is a monosaccharide. It's the sugar in RNA. This other one is beta-glucose. It's another carbohydrate, but it has 6 carbons. 1, 2, 3, 4, 5, 6. And these monosaccharides, they have great solubility. Can you see the reason for that? Look at all these OH groups. All these OH groups are very hydrophilic. And because this is a small hydrophilic molecule, it is very soluble. So glucose is easily transported in the blood, for example. They're fairly stable in their cyclical form, and they can be oxidized in a process called respiration to provide theoretical 4 calories per gram. Now, polysaccharides, they have a variety of functions. They could be structural, or in this case, they could serve as energy storage compounds. So here we can see starch, which is the energy storage carbohydrate for plants. And you can see here how it's made of monomers of glucose. Starch is actually made of 2 polymers, amylose, which is linear like this, and amylopectin, the one shown in this picture. And amylopectin is interesting because it has a branching here. So every 20 to 30 units of glucose, you have this branching, which is very useful because enzymes that digest starch and release this glucose for the plant, they can only remove glucose from the tips of the polymer. So you cannot chop it in half. You can only remove it from the tips. And branching provides more tips, so you can mobilize this glucose faster. For animals and fungi, we use a different polysaccharide. We use glycogen. And if it looks a lot like amylopectin, it's because it really does look a lot like amylopectin. with one difference. Glycogen is more branched, which means you can mobilize glucose even faster from glycogen than from starch. Now, as for the example of our structural polysaccharide, we have cellulose. And this image looks a bit different from what we were seeing before, but still this is a polymer of glucose. So here we have carbon, carbon, all these carbons, and this other carbon here. So we have 1, 2, 3, 4, 5, 6 carbons. And yes, it looks a bit funky. It's not the same regular hexagon. It's twisted, but it's still the same molecule. One difference between cellulose and starch, one very important difference can be seen here. This OH group, this carbon here with the OH group, they're pointing up, but on this one, they're pointing down. This is because the position of this oxygen pointing up, because this glucose is what we call beta glucose. And this bond is what we call a beta 1,4 glycosidic bond. We cannot digest cellulose because the enzymes we have can only act on alpha 1,4 glycosidic bonds. If we had alpha glucose, all the carbon OH here would be pointing up instead of alternating between up and down. So because of this difference in shape, there's a huge difference in function. This form will allow this molecule to be a strong linear molecule, which allows for the formation of these fibers that interact with each other through hydrogen bonds. And all these hydrogen bonds between cellulose molecules will make this whole structure very strong. And now you can see the cellulose fibers made of cellulose molecules. Here we have carbon, carbon OH pointing down, carbon OH pointing up, down, etc. This allows for the formation of these fibers made of many cellulose chains, of many microfibrils, and these fibers will provide strength to the plant's cell wall. Now, some carbohydrates are attached to proteins and they form the structure called glycoproteins in the cell membrane, and they have this role of recognition of signaling between cells. Because remember, cells don't have eyes, they cannot see, they cannot look at each other and say, hey, you are a red blood cell. So they need some form of communication, and in this case, is through the different glycoproteins. This is very useful for our immune system because the immune system has to recognize what belongs to our body and what does not. It has to separate the self from the other because the other could be a threat. And the different glycoproteins we can have in our red blood cells give origins to the 4 blood types. Now, we're not taking into account positive and negative. That's a different story. But for the ABO groups, we have the A-type glycoprotein, we have the B-type glycoprotein, and if a cell has both, well, then it's part of AB. That's because of these little differences. Don't worry, you don't have to know this. You don't have to know the structure. You just have to know that this exists, that A has its own glycoprotein, B has its own glycoprotein. If you have both, you are AB. And if you have none, well, you're type O, which is, ironically, a typo because it's supposed to be A, B, and 0. But if you read A, B, and this oval shape, well, it's supposed to be an O, right? Because letter. This has to be a letter, so AB0 became ABO. Because your immune system has to recognize what is self and what is other, if it identifies a glycoprotein that does not belong to the body, it will attack that cell. So people who have the A glycoprotein, the A group, they can receive blood from people who are in the A group or from people in the group O, because group O does not have a threatening glycoprotein. People who are in the A group, if they receive blood from group B, their immune system will identify the glycoprotein as a threat and it will attack the donor's cells. Same thing for group AB. If a group AB donates blood to group A, You may think, well, that's a good thing, right? Because group A has the A, AB has the A as well. Yes, however, group B also has the B part, and the immune system of the person A will identify the B glycoprotein as a threat and will attack the blood anyway. So A can donate to A or to AB, B can donate to B or to AB. AB can only donate to AB because it's the only group that has both glycoproteins, and O can donate to everyone because it does not have any glycoprotein that can be perceived as a threat. Now for lipids, the thing about lipids is they are very hydrophobic. They do not like water at all. So in this group, we're going to find fats, oils, wax, and some types of hormones, steroid hormones. So testosterone, estrogen, they are steroids, such as cortisol as well, aldosterone. Vitamin D is derived from cholesterol as well. They are all lipids in their nature. And just like we had condensation reactions to make carbohydrate polymers, we also have condensation reactions here with lipids to form phospholipids and triglycerides. This molecule here is a glycerol, and here we have 3 fatty acids. And the condensation reaction will happen between each fatty acid and this hydrogen here from the glycerol. If you take chemistry, you might identify this as esterification. So to make 1 triglyceride, you will actually remove 3 water molecules. Why is it called a triglyceride? Because you use 3 fatty acids and 1 glycerol. Now, if instead of one of these fatty acids, we used a phosphate group, you would have a phospholipid. Phospholipids can be found in the cell membrane, and triglycerides serve as storage of energy. Just as a side note, we have here saturated, monounsaturated, and polyunsaturated fatty acids. This is a saturated fatty acids because it does not contain double bonds between carbons. Now compare this structure to this one. This is a monounsaturated. You see this double bond here? That is what makes it unsaturated. And it's mono because it has only 1. And you see that the shape of the molecule is different. It has a bend here, it has what we call a kink. It bends on this point. And that's important because altering the form, you will alter the function. Now, if saturated has no double bonds and monounsaturated has 1 bond, well, polyunsaturated will have many double bonds. And with many double bonds, you have many kinks, many bends in the molecule. Usually, the more unsaturation, the more fluid the substance will be. Saturated fatty acids will give you solid fat. Monounsaturated and polyunsaturated will give you oils. The more unsaturations your oil has, the more fluid it is. A monounsaturated will be still quite thick. Polyunsaturated is very liquidy, is very fluid. And just so you know, when you read about trans fat, what they mean is the direction here of the carbons. So in this double bond, we have, it's not shown here, but we have a hydrogen atom pointing here and 1 pointing there, 1 carbon here and 1 carbon there. So the carbons are both pointing to the same side, the hydrogens are both pointing to the same side. So because they are to the same, they are cis. But in this case here, it's the same oleic acid, but you have a hydrogen pointing here, a hydrogen pointing there, a carbon down here, and a carbon over there. It is across, they are across the double bond. And because they are across, they are trans isomers, they are trans fatty acids. And the thing about trans fatty acids is because they have a different form, you see, we would need a different enzyme to digest that properly. And we do not have that because naturally, biologically occurring fatty acids, they are all cis. Organisms do not produce trans fatty acids, and because of that, they build up and they cause harmful effects for your health. And where do we find trans fatty acids? In processed food. Now, triglycerides in the adipose tissue, they serve as energy storage, as we mentioned, and thermal insulation. Remember the ring seal that we saw in the water video? If you haven't seen it, take a look at the water video up here. The triglycerides will create a layer for insulating the animal against heat loss. At the same time, it itself is energy storage because 1 gram of lipids will give you 9 kilocalories. Remember that 1 gram of carbohydrates gives you only 4. So it's a much efficient energy storage because with the same mass of lipids, you can have more than double the energy you would conserve with the same mass of carbohydrates. And they are hydrophobic, which means they exert no osmotic pressure. Because they do not attract water, they will cause no influence on osmosis. Now, truth be told, starch and glycogen also do not exert osmotic pressure because they're so big that they are not soluble. But triglycerides still provide the animal with this thermal insulation and a more efficient energy storage. Plus, it can act as shock absorption. We have adipose tissue around our kidneys, for example, for protection, like a bubble wrap around our lip or around our kidneys. We have a capsule of fat. We have fat body behind our eyes, for example. We have a fat body between the muscles in our cheeks. So they provide thermal insulation, energy storage, and mechanical protection. And the phospholipids, they will form bilayers in water due to their amphipathic properties. Amphipathic just means they both like water and they are hydrophobic in one single molecule. So this is a hydrophilic head, and these two are hydrophobic tails. And because a phospholipid is at the same time hydrophilic and hydrophobic, we say they are amphipathic, amphi for both. They have both behaviors, amphipathos. So the consequence is that the hydrophilic heads will be here in contact with water, and the hydrophobic tails will be flipped to the core of the double layer of the membrane. They will be in the center of the membrane, and the membrane will not dissolve in water because the hydrophobic part is hydrophobic. It will not dissolve in water. So it will preserve the integrity of the membrane. This means that hydrophilic things cannot pass through the membrane, but steroids, which are hydrophobic, can pass through the membrane of a cell. So this shape with the hexagons and the pentagon here, this is the skeleton of a steroid molecule. This is the basic frame. And then from that, we can modify and we can have all sorts of molecules. We have cholesterol and we have other hormones. And the two that you have to keep in mind as examples are estrogen and testosterone. And you can see they are very similar with small differences. Testosterone, we will have this CH3 group here that we do not have in the estrogen, plus double bonds here that we do not find in this testosterone. We have a double O here for testosterone and OH for estrogen. You do not need to know how to draw this. You just need to know they're very similar because they're both steroid hormones, and because they're very hydrophobic, they can pass the phospholipid bilayer. So these hormones can freely enter your cell, bind to a receptor, and change the gene expression.